2022 Life Sciences Section Q Biochemistry. Question number 42. Okay, this is a 2 marks question. Which of the following statements about IgA are correct? Okay, we have to identify the correct statements. Option A is it is secreted into colostrum. B, it is transported across the cell by transcytosis. C, its secretion is facilitated by poly-Ig receptor. And D, it primarily exists as a dimer in serum. Okay, so we are all familiar with different types of immunoglobulins, right? So we all know that IgA, so whenever we know that IgG is it has the structure like this. This is the common IgG structure. It has a heavy chain, light chain, and here is the antigen binding. Okay, this is the antigen. It has heavy chain, light chain, right? Here also we have heavy chain, light chain. This is the IgG. When we think about IgA, we all know it is a dimer, right? Everybody knows it is a dimer, right? It is joined by a J chain. IgA is a dimer joined by a J chain. I have structure in the next slide. Okay, so everybody agrees that IgA is a dimer. If any one of you guys have studied the structure of immunoglobulins. Yeah, so here is the structure of immunoglobulin. So we have a monomeric IgA, okay, and then we have a dimeric IgA. Okay, so there are two types of IgA. We have IgA1 and we have a IgA2. Okay, so this these differ in the structure. As you can see, IgA1 is a dimeric, whereas monomeric is IgA2 is a monomeric. Okay, it has similar structure to that of IgG. So we have a variable light chain here. This yellow is a variable light chain, and then we have a heavy chain, right? This is the heavy chain and this is the variable heavy chain, right? We all remember this. Why this is variable? Because this is the variable region. Why? It can vary as per the antigen, right? It is varying according to the antigen, right? If the antigen has a different shape, it will undergo mutation and it will take the complementary shape of that antigen. That is why it is variable. Yeah, very simple immunology. So it is very important that generally we know that IgA is in the dimeric form, okay, but we shouldn't dimeric form. But this is the most important detail, that there are two types of IgA. There is a monomeric IgA and there is a dimeric IgA, and these are found in two different types, and the functions are different. Okay, these are secreted these are secreted for various different functions. Okay, so the dimeric IgA1 is attached to the so that the dimer formation, it is attached by a J chain. Okay, we have a J chain which is also a protein which attaches this to dimer, and then we have a secretory component which it helps in the stabilization of this dimer. Okay, so the functions of this IgA molecule, it is very important. Okay, the functions are they are secreted. Okay, they defend the mucosal surfaces. Okay, we have different mucosal surfaces, like we have different mucosal surfaces like nose, we have throat, we have stomach lining. Okay, so these are secreted on those mucosal surfaces. Okay, our alimentary canal, these are all mucosal surfaces. Okay, from throat to our stomach we have mucosal lining, and on these mucosal surfaces we have IgA secretion. Okay, it is very important to remember IgA is secreted on all the mucosal surfaces along with the primary immunoglobulin, which is IgG. Okay, IgG is a preliminary most predominant form of immunoglobulin, and after that we have IgA, which is secreted in the mucosal surface. Okay, so as we know that it is found in secretions, and it is also present in the serum. Okay, serum as in the blood. It is also circulated in the blood. Circulated in blood. So we have secretory IgA also, and we have serum IgA also. And this is where these are different forms of monomeric and dimeric IgA immunoglobulin exist. Okay. So so in the serum it is the IgA2 is present. IgA2, which is a monomer. Okay, so most of us will make a mistake saying that, yeah, we all know that commonly it is known that IgA is mainly dimeric, but always remember this dimeric form of IgA is only secreted on the mucosal surface. Okay, it is only secreted on the mucosal surfaces. They defend the mucosal surfaces, whereas in the serum, inside the body, where the blood is circulated, the IgA is secreted in form of monomer. Okay, so this you should always remember this. This is a tricky question. This is a 2-mark question. Okay, so coming back to our question. So it is secreted in the colostrum. So in all the mucosal secretion, and also the colostrum. Okay, so as I said, the IgG is the primary secretory immunoglobulin, but all the secretions, which also includes colostrum, IgA is in the colostrum also. So when the babies are feeding, they get the passive immunity, right? This is a example of a passive immunity, right? Natural passive immunity. You guys remember natural examples of natural passive immunity? This type of immunity is transferred to the individual passively, right? Passively as in it is not inside from the environment, or not from the own, it is not made by the body, but it is from the environment of through a different person, entirely different person. That's why it is passive and not active. Active types of immunity are generated within our body, whereas passive are generated outside the body, right? So the IgA is from mother to baby. So from mother to baby is a passive, right? It is from another person, so it is a passive, and it is a natural, right? It is a natural. From another person to a child can is also passive, and artificial is an example of vaccination, right? Babies, many children undergo vaccination. That is from a different person, or it is developed through an antigen, and then it is given, and then it is injected to a child. So it is artificial, made artificially, right? So that is example. Vaccination is an example of artificial passive immunity. But when mother is feeding the baby, the milk contains IgA antibody, and that is example of natural passive immunity. Okay, this is pretty common, right? Basic immunology. It is transported across the cell by transcytosis. Yeah, this is also true. This is also correct. IgA is transported across the cell by transcytosis. Okay, its secretion is facilitated by poly-Ig receptor. So IgA is largely secreted, and its secretion is facilitated by a poly-Ig receptor. This is also one of the correct fact. And option D, it primarily exists as a dimer in serum. Okay, so this option is not correct. So that's why I said this was a tricky question. We all know that IgA, the structure of IgA in the books is always shown as a dimer, right? This is the J chain, and this these are the two variable chains of the IgA. This is a J chain. So in serum, it is a monomer, whereas when it is secreted in the mucosal linings, or it is secreted in form of in the colostrum, it is always a dimer. That time during secretion it is a dimer, but in the serum it exists as a monomer. Okay, in the serum it exists as a monomer. So the correct options are A, B and C. Okay, D is not correct.